Hello, Year 12s. So to start with this lesson, what I would like you to do is I would like you to pair up with someone you haven't paired up before in your biology lesson. And looking at the time, I want you to, for 5 minutes, test yourself on the revision questions you have done so far. You will have a test coming up next week, so this is excellent revision on what you've done so far. After 5 minutes, restart the video. Okay, I hope you feel a real sense of reward getting more and more right the more you practice. Okay, so just a quick reminder from last week, we spoke about starch being made out of amylose and amylopectin. We remembered that amylose tends to be very, very coiled and it is unbranched. And what I would like you to do is go back to your notes and just add in breaks slowly. This is why starch breaks down a lot more slowly. So if you have a unripe banana, you won't get as much energy as quickly because the amylose takes a long time to break down, as opposed to a ripe banana, which is obviously richer in its sugars. And then the second part of starch is the amylopectin. So there we have the 1,6 linkages creating those branches. And we get a picture typically like that, which we have to recall. And I'd like you to add in your notes, these branches can break off quickly when energy is needed. So while you're writing down, that means again, there's a nice mixture of getting some of the alpha glucose very quickly. So that would be from the amylopectin chains and branches, and some are more slowly. That would be the amylose. Okay, so we then spoke about different both being broken down by the enzyme maltase into maltose at different rates. And we mentioned that starch is the main carbohydrate food reserve in plants. So I got you a picture of millet there, which is used to make birdseed. We got wheat there and we got maize at the top. Okay, so starch for storage. Now it's really, really important to link the structure with and its properties with what it's used for. So we said it's very compact, so we can put a lot of starch in a very, very small space. It's easily broken down, and that's important because this supplies glucose needed for respiration. And of course it's insoluble, so that allows it to remain in one place rather than moving around the plant, and it also doesn't affect the water potential or the osmotic processes of the plant. Okay, so you had made your revision cards and hopefully you've shown now how brilliant you are with that. So let's go on to, yep, you got it, cellulose. Okay. Now, looking at this structure you have on the screen, what type of glucose molecule is that? Alpha or beta? And why? Okay, absolutely. So the hydroxide group is at the top. That makes it beta. Remember, beta is the second, B is the second letter of the alphabet, so it's got the two letters, the O and the H at the top. Okay, so this is the key thing. With cellulose, it's made up of thousands of beta glucose units. And these are held together by glycosidic bonds, forming long unbranched chains. Now, what I'd like you to do is listen through to this. You can then go back and pause the video and write down notes if you wish to. Okay, now when you think about cellulose, cellulose is a structural molecule. Okay, so over here we've got some cotton cotton is pure cellulose. So we have a chain of beta glucose, and we don't want any branches. That's going to make it really, really nice and strong, and we also don't want it coiled. Now, when we have lots of different chains, the way we keep these chains joined together is with hydrogen bonds between the chains. That's very, very important. So I've got a length of beta glucose molecules, another length of beta glucose molecules, and that's going to form a long chain. That is why cellulose tends to be in fibers when we put all those chains together. Now, there's something important I want you to notice, because this looks very, very similar to, can you think which other molecule? Aha, I'm hoping you're saying amylose. But there's a difference because we are joining a beta glucose molecule. What happens is for one pair, like we have over here, the glycosidic bond forms at the top. And for the other pair, you can see here how this chain is upside down. The glycosidic bond forms at the bottom. Now, what I would like you to do is I would like you to draw two beta glucose molecules on a piece of rough paper. Then picture how you have to turn it upside down and then join it up. So I want you to work together on this as a class for a couple of minutes. And if you do it correctly, what you're noticing, and this is where I would do a dance for you if I was there, but you don't have the joy. One glycosidic bond is up, one is down. One is up, one is down. One is up, one is down. Okay, as we can see over here, glycosidic bond at the top, at the bottom, at the top, at the bottom. So pause the video for a couple of minutes and see if between you can show how this is joined together from individual beta glucose molecules. When you're ready, restart the video. Okay, now, so it says over here, on condensation, so as the water is removed from the hydroxide and the hydrogen, the glucose units alternate their positions by flipping over, forming a straight chain. Okay, and you can see that by the 6th carbon chain, that's the one that's hanging at the top, sticking out, how that has been turned over. So if I just flip this downwards, you can see how that happens. So alternate monomers are therefore upside down. Make sure that you can communicate that clearly. So when I join this all together, this is what we get, and the red bits here are showing us the hydrogen bonds. Now let's just take a look where that hydrogen bond is taking place, because we have a glycosidic bond over here, and that is joining to the 6th carbon over there. Let's check again. This is where the glycosidic bond is, and that's that carbon that sticks up. That's the 6th carbon over there. And these hydrogen bonds help to keep those chains together, which allows it, as it says over here, these hydrogen bonds result in strong, rigid, and inelastic structures. They are strong because there's lots of hydrogen bonds. Hydrogen bonds for chemists in the group, you would normally say that they are weak, but because there are so many hydrogen bonds, it makes the overall structure stronger. Now, as outstanding biologists, you all know that the cell wall is made out of cellulose for strength and support. And in fact, if you had to look at the cell wall under the microscope, it's actually made of many layers of cellulose, of those beta glucose molecules joined together with hydrogen bonds. So actually, I've got a packet of spaghetti here. If I look at like this is what we call a fiber. So the cell wall is made up of fibers, and you can see all the pieces of spaghetti over there. And then if I pull out a single piece of spaghetti, that is known as a microfibril. And then if I pull out bits of this, I would have those chains of cellulose molecules. That's a very nice way to remember how the structure goes from small to large or from large to small. Okay, before we carry on, don't forget to stick in your diagrams onto your page as well. And if you need the video paused for more time as a group, that is absolutely fine. Okay, let's move on. Okay, so I just shown you the analogy with the spaghetti, and here is another way that this is shown. So you can see there the cell wall made out of cellulose. If I pull that out, we call it a fibril or a fiber. We're going to call it a fibril. We then pull out a microfibril, which is made out of many of those. Okay, so these microfibrils have a great tensile strength because each of these are being held together by those hydrogen bonds, and they interwoven, and that's what allows it to be permeable. Anything can actually go through the spaces. Okay, so they are interwoven and interlinked, and it just allows it to be permeable. It's these fibers that give the plant walls their strength and rigidity, and that allows it to resist osmotic pressure or pulling forces. If you can imagine, a tree has water moving along those cells, often against gravity, and therefore it does need a lot of strength. You learned about that way back in year 7, but now you know why. And the structure also resists digestion by enzymes and the action of microorganisms. So animals have had to really adapt if they want to rely on a diet of cellulose. We are absolutely rubbish at digesting cellulose. In fact, it just gets used as fiber. So we get no nutritional value from eating things like lettuce. And of course, they are insoluble, which is good because you don't want your cell wall dissolving away. Right, for the next bit, we're going to have a look at a video. Now, if this video doesn't work, I want you to play it separately, and I want you to watch very, very carefully what happens to the giraffe as it is eating.